As you know, there are number of chances of DNA mutations, either due to the presence of some mutagenic agents or due to replication errors. So each cell is prone to a large number of mutations. So in this situation, the cell or the organism must have mechanism to reverse these mutations, to repair these mutations by itself. So such mechanisms exist, and the organisms, the cells have number of such mechanisms by which they repair their mutations. The first clue to the existence of an enzyme that catalyzes the direct reversal of DNA damage was reported by Albert Kellner in 1949. He was doing experiments on some bacteria, and in his experiments Kellner first irradiated bacteria with UV light at the doses that killed most of the bacteria. There are some wavelength of bacteria, wavelength of UV, and there is some dose of UV that can kill the bacteria. So he exposed bacteria to that UV, that dose. Then he tested the survivors to isolate the desired mutants, because after that exposure he was expecting most of the bacteria killed, but then he found some of the survivors, and then he tested those survivors so that he may get some desired mutants, because he was working to get mutants. Even though Kellner was very careful in his experimentation, he noticed a great deal of variation in the number of survivors from one experiment to the other. So everybody, when performs experiments, he expects the replication of results, same results in different batches, or even in the same set of experiments. There must be correlation between the experiments, and similarly he was expecting the similar results, but he was surprised to see there was a great deal of variation in the number of survivors. Finally, he found that cells placed in the dark after UV treatment had a very low survival rate, whereas those placed in light had a high survival rate. That means the bacteria which were mutated after UV irradiation, and then those bacteria was placed in light, they survived. That means light has somehow corrected the mutations, while the plates UV treated bacteria were placed in dark. The mutations could not be repaired, and most of the bacteria were killed, and there were less number of survivors in those plates which are placed in dark. Exposure to light thus reverse the UV light's bactericidal effect, because UV light has bactericidal effect due to mutations. If many mutations gather in the genome, the bacteria will die. So this light has reversed this phenomena. Similar phenomena was also observed by Renato Delbecco while studying UV irradiated phage T2. He was working on a bacteriophage which is called T2. Delbecco prepared multiple plates, each containing the same number of UV irradiated phages and sensitive bacteria. So when he was doing experiments, he irradiated phages and then plated these phages in the plates, and he added same number of phages in all the plates. The UV treatment was also same for all the cases, and the sensitive bacteria These are those bacteria which are attacked by this bacteriophage. The number of these sensitive bacteria were also same. So he was expecting that in all the plates the number of survivors should be same. And he then placed the plates in a stack under a fluorescent bulb in the lab. So he put all these plates in a stack, and then he put this stack under a fluorescent bulb which was present in his lab. Each of these stacked plates should have about the same number of plaques. Plaque is a clear zone which is formed on the plate, and it is formed at the site where the bacteria, where the bacteriophage has lysed the bacteria. So the bacteria were killed, lysed, and so a clear zone was formed. So this clear zone is called a plaque. So he was expecting almost same number of plaques in all the plates. However, the plaque number decreased dramatically, going from top to the bottom of the stack. That is, the number of plaques decreased from top to the bottom, because in the bottom the number of plaques was less. Delbecq explained this by proposing that the plates on the top of the stack were exposed to more light from the bulb as compared to the plates on the bottom of the stack, which were less exposed to the light. He tested this hypothesis by exposing some of the plates to more fluorescent light while keeping others in the dark. He had in his mind that the light has in some way repaired the mutations. So he was trying to test this hypothesis. As expected, the number of plaques on the plates exposed to light was much higher than those left in the dark. The number of plaques in the light plates, the plates kept in light, was high. That means when he irradiated phage, the phage got mutated, but and the phage should not be able to attack the bacteria and lyse the bacteria. But when he placed these phages in light, light repaired the mutations in the bacteriophages and making them, enabling them to attack the bacteria. So the number of plaques was more. The bacteria were somehow using visible light to repair the UV damage DNA in the phage. So the bacterial machinery was used to repair the mutations in the phage, and with the help of light. The chemical basis for this light dependent phenomena, which Delbecq called photoreactivation. The Delbecq called this phenomena as photoreactivation, but the chemical basis of this phenomena is remained to be elucidated.